Hello and welcome to New Frontiers in Research and Care. My name is Annie Long and I'm a Community Program Manager with the Parkinson's Foundation here in Georgia. We want to thank our sponsors, Accorda Therapeutics, Amneal, Supernus, formerly known as US World Meds, Kyowa Kirin, AbbVie, and Adamas, who made today's program possible. The Parkinson's Foundation hosts virtual events each Monday, Wednesday, and Friday. To learn more and to register, please visit parkinson.org/pdhealth. You can also visit this page to view archived PD Health at Home programs. Studies show that singing can help maintain strength in the muscles involved in speech and voice in people with Parkinson's. Join us next Wednesday for a fun way to exercise your voice, speech, and swallowing muscles and sing out loud with Parkinson's. To learn more and to register, please visit parkinson.org/pdhealth. And don't forget to register for Moving Day Atlanta, a virtual walk for Parkinson's on Saturday, October 17th. Encourage your friends and family to join your virtual walk team and tune in for inspirational stories, local fitness demos, and so much more. Visit movingdayatlanta.org to get started. The mission of the Parkinson's Foundation is to make lives better for people living with Parkinson's disease by improving care and advancing research today until there's a tomorrow without the disease. In everything we do, we build on the energy, experience, and passion of the global Parkinson's community. We provide free resources, including our website, parkinson.org, educational books, webinars, podcasts, our hospital safety kit called Aware and Care, and our toll-free helpline at 1-800-4PD-INFO, which is staffed by Parkinson's specialists. All right, without further ado, it's now my pleasure to introduce today's program moderator, Paul Tucker. Paul is the past president and current member of the Parkinson's Foundation Georgia Chapter Advisory Board. Paul, take it away.
Good morning, everyone. As Annie just mentioned, my name is Paul Tucker, and I sit on the Advisory Council for the Georgia chapter of the Parkinson's Foundation. I am also, like many of you listening today, a patient living with Parkinson's disease. I've been diagnosed for about 4 years now, and when I was first diagnosed, research wasn't something that I actually gave a lot of thought to. But over the past 4 years, as I've begun to understand more about the disease, learned about the disease, researched more about the disease, I now understand how critical it is to participate in clinical research. And we all need to play our part and get involved to find better treatments for each of us and eventually find a cure, which is obviously what we all want so badly. So as Annie said, one of the major objectives of the Parkinson's Foundation is to make life better for those of us with Parkinson's disease. And one of those factors that goes into that is actually research. And I don't know how many of you know how much research is actually funded by the Parkinson's Foundation, but on an annual basis, the organization funds $10 million to support the most talented minds in research to explore what causes Parkinson's, how to treat it, and ultimately how to cure it. Since 2009, the Parkinson's Outcome Project is the largest ever clinical study of Parkinson's disease with more than 13,000 participants in 5 countries. Through this groundbreaking initiative, Parkinson's Foundation Centers of Excellence track and monitor their care of people with Parkinson's over time. One particular research study that I think is extremely interesting and extremely exciting, I just want to mention to you for a few moments now, is the PD generation program. And I'm going to read some things, so excuse me reading this document, but I want to get this information right so everybody understands what PD generation is about. What it is it's a free genetic testing and genetic counseling for people with Parkinson's disease. Participating and contributing your genetic data, DNA, and blood gives you the opportunity to better manage your disease in the future, help scientists in their journey to advance understanding of Parkinson's disease, improve PD care and research, accelerate enrollment in clinical trials, and aid in the development of improved Parkinson's treatments. For people with PD, genetic testing can let us know if we carry known genes that are linked to the disease. It also gives us a better understanding how those with genetic forms of PD experience symptoms related to Parkinson's and respond to treatment. Scientists can then begin to develop improved treatments and personalized medicine. I think this is a really, really cool initiative that anybody on the phone should actually get involved with. I know I myself signed up, I believe at the end of last year. Initially, the research project was going to be conducted at 6 or 7 centers of excellences that are scattered across the United States. But that has changed. With the advent of COVID, the decision was made to now introduce a virtual factor. So that's the major difference between starting the PD Generation project last year and what we're doing right now. So initially it was done all on site, but now it's changed. So you can actually participate in this genetic counseling and testing at home. And basically, instead of drawing blood, they'll do a cheek swab. And the rest of the program is exactly the same, but it is done virtually. So I encourage you to visit parkinsons.org/pdgeneration. You'll find all the information there about the program. Sign up. I think it's one way that each of us can participate. We can find out a little bit more about ourselves, and hopefully we can aid in the development of treatments that are better in treating our symptoms, and then, as I said earlier, ultimately a cure. So I encourage you all to take a look at that. So now it is my pleasure to introduce our speaker for today, Dr. Dan Huddleston. Dr. Huddleston is a movement disorder neurologist and MRI researcher at Emory University. He is a board certified in adult neurology, and his clinical interests include Parkinson's disease and related conditions. He graduated from Dartmouth College and then completed his medical school training at the Columbia University College of Physicians and Surgeons in New York City. He carried out neurology residency at the Columbia University Medical Center and then came to Emory for a 2-year fellowship in movement disorders. He joined the Emory facility in 2016. So with that said, I appreciate your time and your attention, and I'm going to turn it over now to Dr. Huddleston.
Thank you, Paul. Really appreciate the Parkinson's Foundation and everyone for inviting me to speak today. So I'll be discussing our research, and in particular, I would like to help you to understand, the role the research plays in general and the search for better treatments for Parkinson's disease. And specifically, I'll focus on neuroimaging. This is what my laboratory focuses on, and my team of collaborators, what we do is we use neuroimaging to try and better understand Parkinson's disease biology. But we also want to develop MRI tools that can be used to help improve clinical trials and hopefully lead to the development of better treatments for Parkinson's. So I can tell you a bit about some of the current research that we're doing, and I'd also like to point out a few opportunities for caregivers to find support and to participate in research. So I just have a couple of disclosures. I'm an inventor on some patents that relate to some of our MRI methods. The title of the scientific talk is Imaging Neuromelanin and Iron Pathology in Parkinson's Disease. And I'll explain what I'm talking about there. There are certain more famous aspects of Parkinson's disease biology, things that people may have heard of, dopamine cells dying, something called Lewy bodies, but there are actually a number of different changes that happen in the brain, and some of them we can image very well with MRI. So I'll be getting into that and explaining how we take advantage of these things to try and make some useful advances. So the big picture, why are we doing this? So there have been dozens of neuroprotection studies for Parkinson's disease and other related conditions, and they have so far not succeeded in creating a treatment that actually slows down the process of neurodegeneration, meaning, in Parkinson's disease, the dopamine cells in particular are lost, gradually dying off. And what we would love to have is a drug that stops those dopamine cells from dying off. We infer that would stop the progression of the symptoms because so many of the symptoms are related to that. But an opening question is, we've been trying all kinds of different compounds to treat Parkinson's disease. Why haven't we been able to develop a medicine that accomplishes this? So one reason for this is the way that we design clinical trials. The clinical trials to date have, by and large, relied upon clinical measures. So the kinds of examinations that the Parkinson's patients in the audience will recognize, things their doctors have probably done in their visits, which are looking at their dexterity by looking at their finger taps, evaluating tremor, watching them walk. But the problem with these things is for clinical trials is there are other things that can affect your finger taps. If someone has arthritis, that's going to slow down their finger taps, or if someone has back problems, spine problems, that might affect their walking. So the clinical exam is not the best for determining whether a drug for Parkinson's works or not. It's useful, but it's just not gotten us where we need to get to determine which treatments work well. So how can we address this? The concept of biomarkers is really important, and it's at the core of what we do in the lab, which is to find aspects of the disease that are measurable and that relate to the underlying disease process in Parkinson's disease. So if we can measure something that is really a core component of how Parkinson's happens, well then if we have a drug that stops Parkinson's from progressing, we should see that feature or that component of the disease process stop progressing. So if we can develop biomarkers that are able to do that, that would be very useful and might make our clinical trials more successful. Biomarkers can be any number of things, not just imaging. There's a lot of effort to develop blood markers, spinal fluid markers, skin biopsies, even swabs in the, mentioning swabs for genetic testing. There are efforts to develop biomarkers that way as well. But we like to focus on imaging in our lab because it's really, that's where Parkinson's happens, is in the brain. So that's the only way that we can really get at it. You can't biopsy the brain for this purpose. So imaging is the route that we choose. And within imaging, MRI in particular is a versatile imaging tool. We can see the brain with good spatial resolution, meaning a really detailed picture. And there are different kinds of MRI pulse sequences that we can use to get different types of information, brain structure, and what we focus on a lot are measuring tissue characteristics in the brain. So I was mentioning neuromelanin and iron. So these are substances that can accumulate in the brain. They're either there naturally or they accumulate relating to the disease. And it turns out that we can measure tissue characteristics like neuromelanin content and iron content very well. And I'll explain more about that as we go along here. The name of our laboratory is the Emory Parkinsonism Neuroimaging Laboratory. And I've described this is what we're focusing on. And our goal is to improve clinical trials. So some background to set the stage. By the time that patients have obvious symptoms, their walking is slowing down. Maybe they're not able to use one hand as well to write, for example. Their voice might be getting quieter. Maybe they have a tremor. By the time those symptoms are obvious and patients are coming to the doctor and getting a diagnosis, they've already lost a large percentage of the dopamine neurons that they have in the part of the brain where these changes occur. And so by the time of the appearance of symptoms like that, something like 30 to 50% of the dopamine neurons are already lost in Parkinson's. So one of our goals in this research is to do a good job of detecting Parkinson's disease changes early and hopefully even before the symptoms appear that would lead to a diagnosis. And the ultimate goal with that would be to develop biomarkers and then treatments for prodromal Parkinson's disease to prevent it from ever fully manifesting or fully happening. By the way, if you have questions along the way, please put them into the chat. And at the end, we have a Q&A, and we can go over some of them. So an overview of the research that we'll get into in a bit more detail. So we'll look at different parts of the brain that are damaged in Parkinson's disease. The dopamine cell area is the substantia nigra pars compacta. There's another brain region that we also focus on called locus coeruleus that also has a lot of neurodegeneration in Parkinson's disease. And as I said, the disease characteristics that we'll be talking about with imaging are imaging a loss of this brain pigment, a black pigment called neuromelanin, and the accumulation of iron, particularly in the substantia nigra pars compacta. So we can detect these changes with specific MRI sequences, which I'll go through in more detail. And then we can use the disease features that we extract from this data, this image data, to develop diagnostic tools. So to do a good job of differentiating, okay, this brain is the brain of a Parkinson's patient versus this brain is someone who does not have Parkinson's disease or a neurodegenerative disease. Finally, I'll talk a bit about something called REM sleep behavior disorder, which is a prodromal group of people who have only a sleep disorder, not a bunch of other symptoms, but they actually go on to develop either Parkinson's disease or another related neurodegenerative condition at a high rate. And then finally, some future directions and current research studies and the opportunities for caregivers I mentioned. So the pathology of Parkinson's disease, the most famous pathology or brain changes that you would see if you look at someone's brain who had Parkinson's disease are called Lewy bodies. And these Lewy bodies are clumps of proteins that have aggregated. They have a very characteristic appearance, and they've were first recognized over 100 years ago in 1912, and they were first seen in the dopamine cell area just over 100 years ago in 1919. So they form these round inclusions called Lewy bodies, and then it's important to notice there's a protein in here called alpha-synuclein. If you start looking at research on Parkinson's disease, you'll come across alpha-synuclein quite a bit. The Lewy bodies contain a lot of alpha-synuclein, and there are also Lewy neurites, so this is in the cell body and the main part of the cell, and then the axons and dendrites can also contain clumps of this aggregated alpha-synuclein protein. And these things are really characteristic. Whether they're the root cause of the problem or just a byproduct of what's going on, that is still under debate. And importantly, we don't have a way to image it. We don't have there are PET scans, there are things you can do to image parts of Alzheimer's disease pathology, but we don't have that for alpha-synuclein and Parkinson's disease. So we have to look for other aspects of the disease process. So in the substantia nigra, which is the dopamine cell area, we focus on this a lot because it's really the core location of Parkinson's pathology. If you look at a healthy person's brain, or someone who had passed away who did not have Parkinson's, I should say, if you look in the base of their brain, in the brainstem, at the location where the substantia nigra is, with your naked eye, you can see black stripes that are in that location of the substantia nigra, and that the translation from Latin is the black substance, and that black appearance is from this pigment called neuromelanin, and neuromelanin has a lot to do with Parkinson's biology, and that's a whole lecture in and of itself. But it's been noticed for a very long time. Was first noticed really by Tretyakov, one of the previous slides, in 1919 in the substantia nigra, and then in 1938 a German scientist, Rolf Hassler, was able to show that even within substantia nigra, there's a certain part of it that is especially susceptible to Parkinson's disease. And what you see in a Parkinson's brain is a loss of that black pigment. It is gone. It is just, vanished. And that is because the dopamine cells have died off. And as they die off, this pigment is lost as well. So that is a well-recognized part of Parkinson's pathology and something we would like to image. And that has been confirmed many times over the years that this pattern is really what happens in Parkinson's. So as I mentioned, there are large quantities of this neuromelanin pigment in substantia nigra. The other structure that I mentioned, locus ceruleus, also contains neuromelanin. So an imaging method that can visualize neuromelanin can visualize both substantia nigra, pars compacta, where the dopamine cells are, and locus ceruleus, which makes a different brain chemical, norepinephrine, but they both have this pigment. So there are other nuances about how neuromelanin changes. It changes with aging a bit, but there's a really dramatic effect in Parkinson's disease. So we can see that disease effect. The locus ceruleus, just to spend a moment on it, it is important also because there is dramatic loss of the brain cells in locus ceruleus. It's not quite as famous as the substantia nigra. Our main treatment for Parkinson's is aimed at what's happening in the substantia nigra. Carbidopa levodopa, or Sinemet, is a dopamine replacement therapy that really tries to let the surviving dopamine cells in the substantia nigra compensate for the ones that were lost. And it treats the motor symptoms. People's tremor gets better, their walking gets better, they can speed up their movements. But at the same time, there's neuron loss in locus ceruleus, and it is not fully, it's not proven, it's not fully worked out exactly how the locus ceruleus neurodegeneration relates to the symptoms of Parkinson's, but there's a lot of suggestion that it relates to non-motor symptoms, things like cognitive changes, mood changes, energy changes, blood pressure drops. Those things probably relate to the locus ceruleus neurodegeneration. So in addition to the loss of this pigment, neuromelanin, another important brain change in Parkinson's is the accumulation of the metal iron in the brain. So it turns out that, for example, in brain cells, the furnace is the mitochondria. This is a part of the brain cell that really creates all the energy so that the cell can run. And in the mitochondria, there is iron. The molecules that run the mitochondria contain iron. And then, there's a lot of research suggesting that there are problems with mitochondria. Mitochondria die off in Parkinson's disease. So as they're dying off and those cells are dying off, there's this, release of iron. Some of it actually is sequestered in the neuromelanin, but some of it just forms, elementally in the tissue. So as neurodegeneration progresses in Parkinson's disease, another well-replicated pathologic finding is this accumulation of iron, particularly in substantia nigra also, the dopamine area. And that's been, this was from 1924, so we're also closing in on 100 years of recognizing this issue. So what do we want to do? What do we need to do in order to make useful imaging tools that go after this brain pathology? So we want to be able to study these aspects of Parkinson's disease in living patients, not just in animal models, and in people who have Parkinson's disease. And we want to develop measures of these pathologies and maybe combinations of different measures to make what we call like an MRI signature of Parkinson's changes, so that we can do a better job of diagnosis, do a better job of monitoring the progression of the disease. So MRI sequences to detect this biology are very important as well. So we need specific MRI sequences that can really detect neuromelanin and iron. And we need to be able to do this reproducibly. If we're going to use it as a tool in the clinic or in research. If we do the scan twice, we should be getting the same result. And believe it or not, that one test rules out a lot of the things that we attempt in research. So it has to be reproducible. And ultimately, we want to apply it to these important problems: diagnosis, selecting the right patient for a clinical trial, monitoring the outcome in the clinical trial. Is the drug working or not, or the therapy working or not? So I won't get into the nitty-gritty details of everything about how these things work, but there are two main sequences that I'll focus on here. Neuromelanin sensitive MRI is a rapidly emerging field. It was first reported in 2006, and it's a special kind of MRI sequence that gets nice bright contrast in the substantia nigra and in the locus coeruleus, and allows us to quantify any change, compare healthy individuals to Parkinson's individuals, for example. There are multiple ways of measuring iron with MRI. Magnetic resonance imaging, it stands to reason that iron in the magnetic field is going to do something rather striking, and indeed it does. So one that we have focused on quite a bit, and that I'll show some data from, is R2 star, which is a robust way to measure iron in Parkinson's. So here, without further ado, is our picture of what this contrast looks like. This bright stripe here, one on each side, is substantia nigra pars compacta, where the dopamine cells are, and here it is blown up. And here is some tiny bright dots. Locus coeruleus is a much smaller structure, but you can see that contrast in locus coeruleus as well. These are healthy individuals on top, and in a patient with Parkinson's, the contrast is washed out a bit, and it's really basically vanished here in locus coeruleus. So our lab did research to help demonstrate how does this contrast work. And again, that gets into some details that I won't dwell on, but it was important that we understand how does this work so that we can be convinced we're really imaging neuromelanin. We've also done testing to demonstrate that our methods for measuring the volume of substantia nigra, pars compacta, and locus coeruleus, the dopamine and norepinephrine areas respectively, we've shown that our methods are reproducible. So if I measure the volume of someone's substantia nigra pars compacta, take them out of the scanner, and then put them back in and do the scan again, we get the same result. And so we showed very high reproducibility for both SNC and LC, substantia nigra pars compacta and locus coeruleus. So we also, if we're going to convince ourselves that we're really doing a good job of detecting Parkinson's effects, we need to demonstrate that. So we've done that now in two separate cohorts of patients with Parkinson's disease versus healthy control individuals, and done it on different scanner platforms, different types of scanners, to show that this is really a robust method. And in fact, so cohort 1 was on an older scanner model. Cohort 2 is on a newer scanner model. But we were able to see a significant reduction in the volume of the dopamine cell area, which is substantia nigra. And this is statistically significant in the Parkinson's group, lower than the control group. And the same with the locus coeruleus volume, lower in Parkinson's than controls. And actually the differences were a bit stronger in the newer scanner model, which has, it's a better scanner, has better signal to noise characteristics, it works better. Again, we see very significant reductions in the volume of these structures in Parkinson's compared to controls. We also took advantage of this neuromelanin sensitive MRI method, not only to measure neuromelanin, but to just define where is substantia nigra. That is a big problem too. It sounds simple, but there are a lot of research is done with it's an anatomic expert drawing where they think either substantia nigra is or locus coeruleus is, and as you might expect, not every anatomic expert hand draws the things exactly the same. So we need a standardized method. And so we used the contrast coming from the neuromelanin to make a region of interest that we could use. We have it in what's called standard space, which is sort of like an average brain. And then we can use that tool, that region of interest tool, to accurately select the substantia nigra in anyone. And that is, again, very reproducible. That is something that's done automatically, so there's no operator-dependent drawing part. So we can use that. We call it a mask. We call it a substantia nigra mask. And we can use that to measure other things with MRI. So when we want to look at the iron accumulation, we define the region based on the other contrast that does a good job of defining the region. And then we can look at iron accumulation. So we have done this in a number of different ways, and we show that there are strong disease effects. Accumulation of iron occurs in the substantia nigra in Parkinson's patients, as measured with this approach. They're even, we've got these two contrasts. We've got neuromelanin MRI contrast. We've got iron sensitive MRI contrast. And, we looked at their overlap in the substantia nigra. And that's what this picture is. So we've got, all the colored regions here are where substantia nigra pars compacta is, where the dopamine cells are. The hot colors, yellow and red colors, are where there is overlap between the iron contrast and the neuromelanin contrast. And the take-home message here is, in control individuals, healthy people without Parkinson's, you don't see much overlap. And there's not much iron in substantia nigra pars compacta. In Parkinson's, there's a lot of iron in substantia nigra pars compacta. And that's a really strong effect also, and is a promising biomarker to detect Parkinson's disease. So, there are a number of features that we've identified so far, not just the volume of substantia nigra and locus coeruleus, but the iron content, and then even looking at that overlap region that I was just showing. It happens that this overlap region is in the most vulnerable part of substantia nigra to Parkinson's disease. So we made even a region of interest selection tool for that. Basically we can use that observation to select this area that's most vulnerable to Parkinson's. So we've detected Parkinson's effects in a number of different ways, and as I was mentioning, we would like to define a signature of Parkinson's disease. So to do that, we took these MRI findings, and then having extracted features that we observed to be relevant to Parkinson's disease, we can use machine learning to determine a signature, so a combination of different MRI changes that do a good job, or the best possible job, of differentiating Parkinson's disease from controls. So we create a machine learning classification model, which is what it's called, and we put in MRI features, and we put in some clinical measurements, simple things like age and gender, but also some questionnaires about Parkinson's symptoms. And we put them all into a model and let the machine learning algorithm decide, okay, how do I combine these different things to optimally differentiate, what is Parkinson's and what is not. And so when we did that, the machine learning algorithm it trains on your data set, and then you have a testing data set, and you can see how well it performs on the other data set. But the punchline there, the bottom line, is that when we looked at it with these MRI features and added in some clinical features to the model, we had high accuracy in differentiating Parkinson's from controls. We got up to 94.2%. An important sort of context for that is that there was a recent meta-analysis of Parkinson's disease diagnosis, and unfortunately, the the accuracy of clinical diagnosis of Parkinson's disease is not as high as we would like it to be. And in fact, in this meta-analysis where they combined a lot of, as we say, clinico-pathologic studies where patients were diagnosed and then they came to autopsy to confirm what they really had. In a group of patients who were examined by people who are not specifically movement disorders experts, maybe a general neurologist, maybe even their primary doctor, Parkinson's diagnosis was only about 74% accurate. And then even among movement disorders experts, it was only about 80% accurate. So there's a lot of room for improvement, and also the accuracy of the model here shows that this might be something that takes us to, a better level of accuracy. So we think this can be clinically useful, and we're continuing to develop it, attempting to make a clinical tool with it. So where are we going with this? There are potential applications for this in not only helping doctors to better diagnose Parkinson's, but also to help us when we're designing a clinical trial, might do a better job of selecting who's the right patient who truly does have Parkinson's disease and maybe not one of the other related conditions or a mimic of Parkinson's disease in the trial. We do need to do this in larger groups, in additional cohorts, just to validate it and improve its generalizability so we can really be confident it works in everyone. We want to do a longitudinal study. We've done some longitudinal study already, but we want to do more with it to really fully develop progression markers. That's the other thing that we really need for clinical trials is, some feature or group of features from the MRI image that move over time. So as someone's Parkinson's progresses, we want to see something that is, either going up or going down, so that we can, look at that as a marker of the progression. And then if you have a drug that is going to stop the progression, that, the movement either up or down of the marker should stop. And so we do have some longitudinal work underway currently, and we're looking to get funding to do more. A talk on Parkinson's imaging would not be complete if I didn't spend a moment to mention DAT scan. So the DAT scan it's an FDA approved scan. It's a radionuclide scan, meaning patient receives injection of a radio tracer drug. They have to wait 3 to 6 hours, and then they can get a nuclear medicine scan that will image their dopamine system. And it images in a different way than what neuromelanin sensitive MRI does. But it is useful for differentiating neurodegenerative Parkinsonism from other things. So that it doesn't really tell you specifically does someone have Parkinson's disease, because it doesn't differentiate between the various forms of Parkinsonism. But it can tell you, does someone have Parkinson's disease, or do they have something like essential tremor, which is not a neurodegenerative Parkinsonism. So it definitely has its place. The drawbacks are it's expensive. It takes a long time to administer because you have to give the drug, wait, and then do the scan. And there's also a lot of variability in the quality from one center to the next. And that may be the biggest issue with it because the whole process has to be done just exactly right. So MRI may be something that can bridge some of the gaps there with DAT scan, but it is a useful tool that I order and probably some members of the audience might have had. Here's just an example of the DAT scan. So a normal DAT scan, this is the striatum. So the substantia nigra projects the dopamine cells up to the striatum. This is where the dopamine goes to. And it has kind of this comma appearance, one on each side. And it turns out in Alzheimer's disease, DAT scan is normal. And so you see the comma appearance here. But in a Parkinsonian condition like Parkinson's disease, or here dementia with Lewy bodies, you lose what's called the putamen part of this part of the comma. So then it looks like a period. So the period is the abnormal DAT scan appearance. Finally, I'll spend a moment just to tell you about work we're doing to image prodromal synucleinopathy. So what that means is we would like to image Parkinson's disease and conditions like it, so we call them the synucleinopathies, all the conditions that are like Parkinson's in that they have accumulation of this abnormal protein, alpha-synuclein, that I was mentioning. It's not just Parkinson's. There's dementia with Lewy bodies. There's something else called multiple system atrophy. All of them we lump together and call synucleinopathies. But it turns out there's a sleep disorder, which a lot of people here may be familiar with also. And it's when a patient is calling out in their sleep, acting out their dreams. They may fall out of bed. They have an intense dream and they think they're in a fight or being chased, and they accidentally punch their bed partner in their sleep. That's called REM sleep behavior disorder. It can happen in Parkinson's disease, but sometimes it happens on its own with nothing else happening. And we could call that, well, we call it idiopathic REM sleep behavior disorder. It's just isolated. And it turns out that group of patients goes on to develop either Parkinson's disease or one of the other related conditions, one of the other synucleinopathies, at a high rate. So if you can identify a group of patients with just a sleep disorder, it's a prodromal group. It's a group that likely has early Parkinson's disease or related condition, and it's good to study to develop biomarkers, and then hopefully we can develop drugs that can keep those people from going on to get Parkinson's disease or one of the other conditions. And so here's kind of a graphic representation of this. There's a point here where symptoms are coming on. So here's your red severity of motor symptoms line, and after you cross the threshold, people actually get diagnosed. But that's pretty late in the game. Neurodegeneration started a long time before, and we've been losing dopamine cells for quite a while there. And so indicators of that early process, the most specific ones are REM sleep behavior disorder I was just talking about, and loss of smell. And there are also some more non-specific things like mood changes. So we are studying these patients right now. We have a study to clinically characterize them, and we are doing MRI on them. We're part of a consortium of 10 centers and growing around the country to collect a large group of these patients. And we have a small grant funded and doing some initial studies, but we have another one that's under review right now, hopefully to expand that. So, it's relevant to mention that locus coeruleus and substantia nigra are affected in this prodromal stage, and we have some preliminary findings showing that we can actually detect those disease effects even in the prodromal stage. So this is the consortium for REM sleep behavior disorder that I was talking about, the North American Prodromal Synucleinopathy Consortium, which has the cute acronym of NAPS because it's a sleep disorder. Our current study is it's an NIH-funded study in R34 that really got us going, got us off the ground. I think we're now more up to about 245, not 215. And our site leads the imaging component for this study. And then the pending grant is the NAPS2 consortium. And so this is one that is going to be a much more detailed study. It will include imaging. I'm sort of doing the imaging on the side, but this one will hopefully really develop our research for early detection of Parkinson's disease and related conditions. We also have a study going on to do differential diagnosis, differentiate Parkinson's from other conditions. There are atypical Parkinsonisms that are often confused with Parkinson's disease, something called multiple system atrophy, something called progressive supranuclear palsy. We're using these same MRI methods and others to differentiate these conditions. And we also have a Lewy Body Dementia Association Research Center of Excellence at Emory. So this is relevant to Parkinson's disease. So the Lewy body dementias are Parkinson's disease dementia and dementia with Lewy bodies. And so Lewy body dementia is a category. So people with very advanced Parkinson's disease tend to have more cognitive impairment and develop dementia. So we have a center that is really about, first of all, making sure patients connect with clinical care who need it, but also part of a clinical trial consortium. We need more candidate drugs for these things, so that's been the issue so far, but there are things coming down the pike, and we hope to offer that as well. I would put in my plug for caregivers here. Part of what we do through this center is we're just kicking off a virtual caregiver support group for patients with Lewy body dementias. So if someone has Parkinson's disease and they've developed cognitive problems with that, this is to help support the caregivers for those patients. So do get in touch with us, and I have Kelsey's email here. Kelsey is our lab research coordinator and also Paul's daughter, so just reach out to her if you're interested in that as well. And finally, we do have an APDA center for research where we're imaging Parkinson's dementia and DLB. All of these studies are recruiting. So we're recruiting patients with Parkinson's disease without cognitive impairment, patients with Parkinson's disease with cognitive impairment, patients with other kinds of Parkinsonism, all kinds of Parkinsonism. So, if you or someone you know is interested in this, and I would also say caregivers, people who don't have any of these things but want to be involved in the research, we always need individuals who can be in our control group and who can help us with research that way. So do get in touch with us if you're interested, and I thank you for your attention. I want to acknowledge our funders and our lab. Thank you.
Okay, thanks, Dan. That was great information, and I think you did a really fantastic job taking a very complex topic and distilling it down to understandable means for many of us in the audience, so I appreciate that. Thanks very much. So we're going to take a few questions now from those of you who have sent some questions in through chat. I would encourage you, if you have additional questions, to keep sending them in. So I'm going to go ahead and read some of these questions, and Dan will do his best to answer them for you. So the first question: I'm a non-manifesting LRRK2 G2019S mutation carrier. Do you have future plans to open your Emory MRI studies to include the at-risk population?
That would be great, and that's interesting that you have that information. And I don't know if that may have come from like 23andMe. They're doing that, I know, but I'm certainly interested in that. I think the main thing is we have to develop the resources to do studies like that. If you could reach out to Kelsey, I would like to, if you're interested, I would like to keep track of that. If we can come up with like a group of people who we know have this, then it's a lot easier to apply for funds to really get a research study going. And I can do a few things kind of one-off, but to really do a good job, we need to get a lot of people. But thank you for bringing that up. That's a great question. And yeah, if you're interested, get in touch with Kelsey and we'll keep track.
Okay, thanks, Dan. Question number 2. I participated in PET scans and functional MRIs in Vancouver several years ago. I was given a CD of my MRI images before I left. I really appreciated that. Do you, or would you consider, giving participants a copy of their personal MRI images, albeit without interpretation, at the conclusion of your studies?
Yes, if the important thing is that we know in advance. It's fairly straightforward to do if you tell us that's something you're interested in before we do the scan, because we would typically want to burn the disc at that time. It gets a little more complicated if we have to go back later and do it. But yes, that is something that we can do.
Okay, thanks. Question number 3. Is the same MRI used in sleep DBS?
is the same MRI used in sleep DBS. So do you mean, are you talking about intraoperative DBS? I know this is kind of hard for the chat format, but so I'm going to conjecture that what you're talking about is intraoperative MRI that's used for deep brain stimulation surgery. So no, this is a research. This is a 3 Tesla MRI, which is a stronger MRI than is used in most clinical MRIs. Now there are some clinical MRIs that are 3 Tesla now, and we have a couple at Emory. But if you go to a community hospital, it's usually 1.5. If you're having intraoperative DBS MRI, it's usually 1.5. So that field strength actually matters a lot relating to what you can do with MRI. So it is different than that. However, intraoperative MRI is really an interesting area, untapped area for research. So I think there probably will be things that are interesting to do with that also.
Okay, great. Another question from the field. I guess if someone is interested in participating in your studies, Dan, what is involved in a typical study visit and how long does one visit take?
Great question. So the visit consists of sort of a clinical characterization part where, first of all, we'll send you some questionnaires and things ahead of time that you can do at home. And then you come in and we do things like I will examine you and do kind of a similar exam to what patients with Parkinson's often have with their doctors, with a few more things added, and then various other kinds of questionnaires and assessments, asking questions. We check your blood pressure, lying and standing. We get a blood draw. That whole part takes something like 3 hours to do. And then we go to the research MRI scanner, and that is an hour MRI scan. There's a little bit of transit time in between. So the whole process is something like 4.5 to 5 hours for the whole visit. It's a one-day visit for the things that we're doing currently. I was mentioning one thing that we're trying to do is do more longitudinal imaging and applying for funding to do that. So certainly people who are interested, we'd like to bring them back in the future once we're able to.
Okay, I think 1 or 2 more questions will do it. So next question, Dan, how important is diverse participation and how do you accomplish that?
Very important. If you want to have generalizable results, I was talking about you want this to be a set of tools that works for everyone, not just for one little group and then can't use it for everyone. So certainly racial diversity is really important. We have a more racially diverse group here at Emory than in a lot of academic centers. But we still suffer from some of the same problems with academic research. Academic research tends to attract people with higher socioeconomic status, and recruiting some racial minorities. Like we could certainly stand to have more racial diversity and ethnic diversity. So I think it's getting the word out to the community. We try to, we compensate people for their time. We try to make it not a burden, to participate in the research. There's a research incentive paid for all the visits, which is somewhere between $50 and $100 depending on the visit. So we try to make it easier for people, but just getting the word out is helpful. And if you know of people who might be interested, who fit the bill, either control or as patients, certainly let us know.
Okay, question number 6. MRI machines are widely available. What would it take, by way of cost, training, and so on, to adapt current methods to include neuromelanin and iron detection on a wider scale in the US once it is validated?
It's a great question. And that's a core question to scaling this up. So I was mentioning, one of the issues, we want to do 3 Tesla MRI. So most community MRIs are 1.5 Tesla, but the prevalence of 3T MRI is rapidly increasing because you get a much better picture from it. And there's also sort of a need for the, to borrow from I guess the tech guys, the killer app, so like something that really makes the 3T MRI much more valuable. And so the first step would be to get this out to all the 3T MRIs, which would be a lot of academic centers, and even a lot of larger clinical centers have 3T MRIs. The sequence that we have is written for Siemens scanners, but it's easily adapted for other brands, and actually Siemens is the biggest vendor for this. So, you know, we currently have actually a grant to develop this as a clinical tool, to have an image processing platform that automates everything and could take in data from around the country. And we're basically, you know, we are finishing the prototype for that right now. We need to get FDA clearance. We need to do a couple more things, but those are in process currently. So, you know, my hope is that we have something FDA cleared that can be done in kind of a prototype basis at 3T centers around the country, you know, in the next, you know, 12 to 18 months. So that's our goal. And I think it's very achievable. You know, the good thing about MRI is safe. This is a non-contrast. You know, we're not injecting MRI contrast. You don't have to inject anything. So there's not a lot of barriers to it. It's cheaper than DAT scan. So I think all the ingredients are there, and we're just trying to, you know, move it forward and make it happen.
Okay, one final question, and then we'll call it a day. Why don't studies use participants who have PD in their family and diagnosed REM disorder if they are under 50 years of age, especially if they are willing to participate?
We do. So we don't have an age limitation for Parkinson's disease, and we've imaged people, I think, as young as about 40. You know, it starts to get increasingly rare younger than that, but it certainly happens. You know, I've had patients in their 20s with Parkinson's disease. So we are interested in that. And now there are different, like, depending on what researchers are doing, you know, there's a differentiation between early onset Parkinson's disease and later onset Parkinson's disease. And usually the arbitrary cutoff is 51. So a lot of the genetic PD researchers look at early onset Parkinson's because it tends to be more often associated with the common genetic causes. So sometimes people are really trying to target that, you know, target the early onset or target later onset. So there can be scientific reasons that make sense for that, but that's not really part of what we're doing at the moment. So if you're under 51 and interested in research, do let us know.
Okay, guys, I think that'll do it for today. We're right on time. So I want to thank Dan again for his participation. And I want to thank all of you for joining the call today. I'd like to take a minute and thank our sponsors once again: Accorda Therapeutics, Amneal, Supernus, Kiowa Keem, AbbVie, and Adamus, who made today's program possible. I just also want to mention to you guys that the recording and any questions perhaps that we didn't get to via email, as well as the slides, will be sent to you guys via email, so look out for that. We do hope to see you at one of our upcoming events. For information on our national programs, just go to parkinsons.org/pdhealth. You will also receive a short survey by email after today's program. Please take a minute to complete the survey. We'd love to know what you thought of and what else you'd like to learn about. We will also share a recording of today's session once it's available, as I already said. So just from my own personal perspective, I just want to throw a couple of thoughts out there. I would encourage you, as I said when I initially started speaking, I would very much encourage you to check out the PD Generation program. Just go to parkinson.org, PD Generation, and you'll find out all kinds of information about how you can participate. I encourage you to participate because there's really nothing that we have to do. In the past, as I said, you had to do it on site. Now the foundation has made it so easy for us. You can just basically do it virtually at home through some telemedicine appointments and things like that. So check that out. Also, if you're interested in participating in some of Dan and Kelsey's studies, just fire off an email to Kelsey. Her email is ktuck, that's KTUCKE3 @emory.edu. And I think that'll call it a day for now. Just one final thing. Whether you're interested in keeping things the way they are in the United States or changing them, it's your responsibility to vote. So have a great day, guys, and we'll see you again. Bye-bye.
Thank you.